Well, I have given a green light to start. So we're going to start the opening lecture of the 5th edition of the Molecular and Cellular Integrative Biology Master of the National Research Council, the Spanish National Research Council, and the International University Menendez-Pelayo. The opening lecture will be given by Professor Bruce Alberts, Chancellor's Leadership Chair in Biochemistry and Biophysics for Science and Education at the University of California in San Francisco. So good morning, San Francisco, where it is currently 8 o'clock in the morning, and good afternoon to those persons who are following us from Madrid or elsewhere in Central European Time, currently 5 p.m., or from the UK and Portugal, currently 4 p.m. My name is Miguel Peñalba, and I will act as chair of this inaugural lecture of the 5th version of the master course on Molecular and Cellular Integrative Biology. On behalf of the Spanish National Research Council and the International University Menendez-Pelayo, and of the Margarita Salas Biological Research Center, where this event is physically being held, I would like to express our deep gratitude for your willingness, Professor Alberts, to be the keynote speaker in this opening session. I reckon that Dr. Germán Rivas, the director of this master, chose me to moderate the ceremony because he's well aware of the strong influence that you had on my scientific career. When I started my PhD working with bacteriophage 529, I was offered 3 different topics. One was the morphogenesis of the viral particle, the second was the transcriptional program induced by the virus in the bacterial host, and the third was DNA replication, specifically the initiation of DNA replication at the ends of the linear DNA molecule, which is the chromosome of the bacteriophage. DNA replication was actually the one I chose after reading an influential review that I have here with me. I don't know whether you can see it. It's a review written by Bruce Alberts and Rolf Stenglach and was published in Nature in 1977. Shortly after I finished my PhD and perusing the shelves of a bookstore in London, I found a textbook, which I immediately bought, which is this, The Molecular Biology of the Cells by Alberts et al., and which has been my bedside textbook ever since. I strongly recommend to young scientists to check their voracity to read the latest paper in the latest top-tier journal published, and to devote some time to read a classical textbook, because they could find by themselves how often the community of molecular biologists are ready to reinvent the wheel and to use hyperbole. So it is important to read the classical textbooks. The third source of influence of Dr. Alberts on my career was the coining of the concept of the cell as a collection of molecular machines. And I will stop here because I don't want to make a spoiler, because the title of Dr. Alberts' conference is actually The Cell as a Collection of Protein Machines, Preparing the Next Generation of Molecular Cell Biologists. I'm passing on the microphone, the virtual microphone, of course, to Mateo and Ana, 2 students of the 4th edition of the Master, who will summarize Dr. Alberts' trajectory before the conference itself. And after the conference, the students will have the chance of addressing their questions to Dr. Alberts. And lastly, if time permits, we will consider a couple of questions selected from attendants that are following us live through the CSIC YouTube channel. Finally, Dr. Germán Rivas, the director of the Master, will close the ceremony. Thank you very much. And then for Ana, you have the microphone.
Hello. In the first place, we would like to tell you thank you very much for being here in this inauguration of the 5th edition of the Master. And it's a great pleasure and honor for all of us, and especially us students. Before starting with the presentation, when Mateo and I were researching about Professor Alberts' career and everything that he has done, we came upon an interesting bit of information, which is actually that Professor Alberts didn't take biology until he went to college, and because he signed up late for biology, so he got an amateur license in radio, and we thought this was quite interesting because the whole talk is going to be about biology. So, well, then of course he took biology and he graduated from Harvard in 1960 in biochemical sciences. And for his graduate degree, he was planning to solve the genetic code. He then went on to get his PhD, also from Harvard, And his initial idea was to unravel the mechanism behind double-stranded DNA replication, although he then changed the theme. He went on to do his postdoc in Geneva, Switzerland, before returning to the United States, where he spent quite a few years at Princeton University. And then in the late 70s and early 80s, he was approached by James Watson to write a textbook. And that is the mythical textbook we all know very well because we've all used it to study during our education. And it's Molecular Biology of the Cell, which constitutes one of the pillars, I think, of all molecular biology sciences. And this is a work which is in continuous evolution, which has many editions, and he's still working on it. So, yes.
So also in the late 70s, then Dr. Albert became a professor and vice chairman of the Department of Biochemistry and Biophysics in the University of California, San Francisco, where he currently holds the Chancellor's Leadership Chair in Biochemistry and Biophysics for Science and Education, as Miguel said. He then served as president of the US National Academy of Sciences for 2 terms, spanning 12 years, as well as editor-in-chief of Science magazine from 2008 to 2013. During that period, he was appointed by President Obama as one of the first 3 US science envoys. For all of his achievements, he has received at least 75 honors and awards, among them countless honorary doctorates of science from universities all over the world. Professor Albert was granted the US National Medal of Science in 2014 and the Lasker Koshland Special Achievement Award in Medical Science in 2016. Throughout his entire career, he has been focused on science education and scientific literacy, trying to raise awareness of science as a way of knowing among the general public and emphasizing the role of scientific evidence as a motor of decision-making. Once again, we would like to thank Professor Alberts for being with us today.
Thank you. The students are impressive. You did all that research. You were, you're going to be professors or maybe even university presidents someday. I'm very impressed. All right, I always love talking to students. As you'll hear, there's so much that we need you to do in science. I really enjoy trying to stimulate students to be more ambitious and solve harder problems. And hopefully you'll have some time to discuss that today. I will try to start my slides. Let's see, I gotta get this. Is that working? Can you see slides?
Yes, we do. We do see the slides.
Okay,
Yeah, everything is okay.
good. All right, great. Let me know if it stops because my internet is not wonderful. So if anything goes wrong, please say something. All right, so I was given this title, and it's a fine title. I'm going to talk about 4 different things, and here's the order. My life in science, which involved the biochemistry of protein machines. We didn't know that when we started. Secondly, writing a textbook and what you learn, which is how little we really understand. Although we have all kinds of knowledge, how little we really understand about fundamental problems about life in the cell, which leads me to the need for much more fundamental research. And finally, just address the students, talk a little bit about how I advise students to think about what to do in their life as a scientist. All right, we'll start with part 1. So I was a high school student when this Watson and Crick structure for DNA came out in 1953. And it really started the whole revolution in what we now call molecular biology. before that time, it was very hard to really think about how cells work. So they really solved the mystery of heredity, but they didn't know anything about the machinery that enables a DNA sequence to be copied from one generation to the next. They of course proposed the right idea. That's what was revolutionary. So I started as a graduate student in 1961, and I was fascinated by the mystery of how you could replicate DNA. And at that time already, Arthur Kornberg had discovered the enzyme called DNA polymerase, which we thought was the only protein that was needed to replicate DNA. And there was a problem, however. It only would work on DNA that was single-stranded. And of course, we knew that DNA inside that cell was a double helix. And so the question was, how could the DNA polymerase work on a double-stranded DNA molecule inside the cell? That was the fundamental wrong assumption that there was only one protein involved. But secondly, the way we thought about cells also was that every RNA and protein molecule inside the cell was bouncing around at high concentration inside the cell. And because we knew how rapidly molecules moved, in fact, I came from a physical chemistry laboratory, and we all thought that you didn't need any organization inside the cell. That every time you added a nucleotide to the end of a growing DNA chain, you'd get a new DNA polymerase molecule adding and then falling off, and then the next DNA polymerase would come, and so on. So we had a very simple idea that basically the inside of a cell was just like a inside of a test tube. So then I had a very dismal career as a graduate student. I made my own theory. We were all inspired by this theoretical biology they had done. They had broken open this heredity problem. So I made a theory. I got an A on a paper for a course in proposing a model for DNA replication where a DNA polymerase might be able to replicate DNA by itself. And of course, my theory was wrong. I spent a lot of time doing experiments to see if it was right, and all my no answers didn't impress anybody. Nevertheless, eventually, after 5 years, my professor thought I was ready to get my PhD, and I walked into my committee meeting with my airline tickets, to leave the next week with my small little family, my wife and one baby, to go to Switzerland for my for my postdoc, and they failed me. They said they didn't like what I had done, and I had to do 6 more months of work before I finally got my PhD. But for students, this is a very important lesson. Failure is a great learning experience. I think if I hadn't failed my thesis, I would have been much less thoughtful as a scientist. I spent at least a month trying to figure out what I had done wrong and whether I could really do science, and try to think about how to go forward so I didn't make the same kind of mistake again. And I actually given talks on the importance of failure, because we don't learn from our successes. We learn from our failures. And older people, your professors, have made many failures, and that's why they're able to be wise advisors. They've learned from their failures. Now, there are people who don't learn from their failures, and they never become professors. So at any rate, learning from failures is important. So you're not to be discouraged by failing. Everybody fails. Take it as a learning opportunity. So what I learned was that strategy is everything in science. And I had a very poor strategy for my PhD. And I also decided that theoretical biology was not going to work. You had to do experiments to figure it out. So I moved. The other thing that happened is I finally did get to Geneva. where I did 1 year as a postdoc, and I suddenly discovered that my whole hypothesis was wrong about DNA polymerase replicating DNA by itself, because in Geneva, I discovered bacteriophage T4. It had more than 100 different genes known from genetics, no biochemistry. But here was a striking fact. I hadn't read, right in the middle of my thesis, the paper was published that showed that my whole hypothesis was wrong. It was a paper in a genetics journal. And it really demonstrated that for bacteriophage T4, it took many different genes to replicate DNA. So another lesson is to read very widely in the scientific literature, which I had failed to do. So there were 7 different genes. They were given these numbers because nobody knew what they did. 32, 41, 43, 44, so on. One of them, by the time I got to Geneva in 1965, one of them, gene 43, had been shown to produce the T4 bacteriophage DNA polymerase enzyme. But the genetics showed there were 6 additional proteins needed for any replication of the T4 chromosome when the virus infected E. coli. And so it was absolutely shockingly clear as soon as I learned this that DNA replication must involve at least 7 proteins and be much more complicated than anyone had imagined. So, given that fact, how do you produce a strategy to try to solve the problem? So since I had failed my thesis, I thought very carefully about that. And I said I would develop a new method to try to isolate all the proteins that T4 made that bound to DNA. And among those proteins would be I thought, the proteins that were needed for DNA replication. And I could use the genetics, the mutants identified by genetics, to identify those proteins and purify them. So I spent a year in Geneva developing this method, which turned out to be called DNA Cellulose Chromatography, where you could stick a high concentration of single-stranded or double-stranded DNA on cellulose. And I found that many proteins in a T4 bacteriophage infected cell bound to this column and could be eluted with salt. And so then you test with different mutant viruses to see if one was missing, and that would be all you need to start purifying that protein as a biochemist. So after 1 year in Geneva, I started my old lab at Princeton. I was 28 years old. That's when we used to start labs in the United States. That's another whole lecture I'm not talking about today. We needed to get back to something like that. So the students and I focused on purifying each of these proteins. Each student got a different protein. Here's a picture of my lab in 1969, 2 years after I got there, or some 3 years after I got there. Many of these were undergraduates. There were 3 undergraduates and 3 graduate students, and a research assistant. And the dog was really not in my lab. It was just a stunt. All right, so our dream was to get double-stranded DNA to replicate someday in a test tube by mixing the 7 proteins together with purified DNA. And it finally happened in 1975. So it took a total of 9 years to get it to work. And that's something to remember. That, you got to be patient and keep at it. And it was not just my lab. There were other labs working on this problem, but together we came up with the correct model for DNA replication. As everybody knows, the 2 strands in the DNA helix run in opposite directions. So DNA polymerase, which runs in only one direction, has a complicated time replicating it. And it turns out that 2 DNA polymerases work at the same time, one making DNA backwards on what's called the lagging strand, and the other polymerase goes continuously on what's called the leading strand. And there are all these other proteins that are involved in clamping the polymerase down and starting the chain on the lagging strand and so on. You read about that in a textbook. So I'm going to show you my favorite movie because this, was a mystery that we worked on for so long, and nobody would ever have predicted anything this complicated. If anybody had made a theory that DNA replicated this way when we started, they would have thought they were crazy. Making one strand as a series of short pieces seemed like a crazy idea. It would never work, but that's in fact how it works. So this little movie from our textbook actually was made at Cold Spring Harbor to celebrate the 50th anniversary of the discovery of the DNA double helix. And the proteins here are moving at the same rate as human DNA replicates inside your own cells. So I'll just play this little video.
Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You're looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So that is an amazing movie. And it shows how complicated the chemistry of life is. It's by far the most sophisticated chemistry known. So today, you could do much better than we did. We had 7 proteins. To get eukaryotic DNA replication to fire off replication forks, it requires 42 polypeptides instead of 7. This is a wonderful paper. We have better technology today that made it easier to do this, but it was important to have the kind of model that I and others developed in simpler systems to guide this kind of work. So one of my major points is that starting with the simplest system you can is a shortcut to understanding humans. So here's a sort of a summary from this paper. Here's how the 42 polypeptides create 2 replication forks heading off in opposite directions from a replication origin in a yeast cell, which is the same as what happens in a human cell. But this is a wonderful accomplishment, but it's only perhaps 1/10 of the proteins that are critical to DNA replication and DNA repair, the processes that are critical for maintaining our DNA sequences. And we don't have this kind of detail for the rest of it. So I just want to make the point that understanding these details from how protein machines work in DNA replication and DNA repair will be critical to improving human health, as well as, of course, for understanding how we work. So I'll just give you one example. Because cancer requires that 5 or perhaps 10 mutations accumulate in one single clone of cells before the cell becomes dangerous. Tumor progression selects for hypermutability often early on in the process. And therefore different tumors will by chance require a very different defect in either DNA repair or DNA replication that makes their mutation rate abnormally high. And there's a great potential in exploiting each particular defect to eradicate a cancer, but of course we have to know what that defect is, and that's what we call personalized medicine these days. And as a proof of this concept, for certain kinds of hypermutability caused by BRCA1 and other proteins in that pathway, therapy has been improved using poly-ADP ribose polymerase inhibitors that actually selectively kills those kinds of cells by making them super mutable and so mutable that they die. And that's, you know, if we knew everything about replication and repair, we could probably do that for almost every tumor. So 2 lessons learned in the last 40 years. First of all, because of evolution, there are remarkable homologies between living things. Therefore, use model organisms to reveal mechanisms wherever possible and then build off of that to understand humans. Secondly, nearly all cell processes will be driven by complexes of proteins, usually 10 to 20 proteins organized as a protein machine. They all work differently, but you see how elegant that one machine is, and they're also, the other ones are also elegant. And of course, to make a machine that's effective, you need ordered protein movements driven by some kind of energetic process. And usually it's the energy of ATP hydrolysis, but it could be a ion gradient, other things. And this, just like the machines we know in everyday life being driven by electricity, ordered movements are the key to making a machine work. And finally, all these processes will be based, like DNA replication was, on elegant mechanisms that are too complex to predict. Okay, so here's a good challenge for the next generation of biochemists, which I hope are some of you. First of all, obtain the information needed to accurately describe the mechanism of every type of machine in a cell. And I'm going to come to some other challenges shortly, but for this one, you need to reconstitute with purified proteins these many hundreds of protein machines from the, and then only in that way can you really manipulate the concentrations and order of addition of reagents, so you could decipher the detailed chemistry of how the machine works. And of course, nowadays you could also do lots of structural studies. So you know, what we're doing today is getting huge amounts of data on proteins and their interactions and their structures, and we know the components of huge numbers of protein machines, but we don't know, for most of them, how they actually work. Of course, there are some that you've been really well studied, like the ribosome, for example, but most protein machines, most of the, say, DNA repair protein machines are not well studied in this, well enough studied in this way. Okay, now the part 2. Many fundamental mysteries remain about the even the simplest living cells. And here's a challenge for young people I'll get to very soon. I've been writing textbooks for 42 years, and I was doing it yesterday again. So I have to stop pretty soon. I'm getting too old for this. Anyway, how did we start writing a textbook? Suddenly I was sitting in my office in 1978 in California. I got a call from Jim Watson, who I knew because he was a professor at Harvard when I was there. He was then 50 years old instead of 25 in this photograph. He said, well, I got a great idea, and it was a great idea. Now's the time to bring together 2 disparate fields, molecular biology, which came out of, you know, Max Delbruck and a bunch of physicists thinking about model organisms and T4 bacteriophage and E. coli and others. But anyway, that field, which I came out of, and another field at that time was called cell biology that I had barely heard of. It was a field that came out of light and electron microscopy, and it had defined all these parts of a cell like the endoplasmic reticulum, which I had never heard of. And Jim said that now is the time to write a textbook to try to bring these 2 fields together, and he was absolutely right. What he was wrong about was how long it was going to take. He got us to do this because he said it would take only 1 month next summer and another month the next summer. So that was way off. So this is the book that was waved around shortly. Proofing this book was very much harder than we expected. In fact, we almost quit at one point because we realized how hard it was going to be. My friend Keith Roberts, who does all the drawings, he's a wonderful asset to this book. kept track of the time. He's very organized. We spent more than a year's worth of 12-hour days sitting together usually over Christmas, Thanksgiving, all kinds of holidays, in addition to the summer. This is a picture of the first set of authors crossing Abbey Road. This is the Abbey Road recording studio. It happens to be next to the house in London where we've worked a lot on the book because most of the other authors were from the UK. And the major point I want to make is each time we write a new edition, we're humbled by how much we still don't know. And for that reason, in this last edition, at the end of every chapter, we added some of these for students, just to make it clear that although textbooks try to explain what we know, there's a huge amount that we don't know, and we need young people to become scientists and solve these problems. So I'm just going to give you two examples quickly. First is the recognition that extensive sets of scaffolds produce biochemical subcompartments in the cell without requiring a membrane. And most recently, that many of these scaffolds create larger phase-separated liquid compartments. And second, the recognition that positive and negative feedback loops underlie nearly all cell chemistry, creating complex networks of interactions, and these give rise to so-called emergent properties that really are at the heart of life. Emergent properties are something that physicists defined in the 1940s, but it's a general fact that when you combine enough complex things in complicated ways, new properties come that can't be predicted from any of the parts. So here's scaffold proteins. This is a figure from our textbook. Scaffold protein is a protein with binding sites for reactants, the things that molecules are going to react together. They're connected by flexible loops. So what you're doing is you're increasing the collision rates for proteins that are going to react together to create products, proteins and other molecules. And the other thing about scaffold proteins, they're almost always located in specific parts of the cell so that the reaction occurs where it's needed. Here's the simplest example. The first one I ever heard of was from Tom Pollard's work, was studying the polymerization of actin. Actin, of course, is a major cytoskeletal protein. The filament would go down through, the floor of the building, many long red filament. We're seeing the growing positive end, and the growth is catalyzed in this case by a molecule called a formin. And Tom's lab discovered that the growth of this filament in a test tube would go faster than the so-called diffusion controlled rate. Diffusion control is a physical chemical term that you calculate how fast actin monomers are colliding with the end of the actin filament. And of course, it can't go faster than that because every collision then would have to, the fastest you could go is when every collision creates a reaction. And the way it could go faster than so-called diffusion control is because it has a scaffold, the whiskers on the formin, actually the formin, which have a loading zone for actin filaments and cause a much more rapid collision of actin with the end than would be predicted from random association if the cell was not organized. So there are also now RNA scaffolds. Some of the link RNAs are like this, not well studied, most of them. Scaffolds form what we call biochemical factories inside the cell. They use much of the principles of phase separation, and we call them intracellular condensates now. This is an old picture before we knew anything about that, of a cell nucleus. Transcription is going on in the red areas, replication in the green areas. You see there are concentrated mixtures of proteins involved in each one. And those only form when you have replication occurring, and then they go away. And likewise for transcription. So these are transient organized components. Intercellular condensates help to form, now we know, help to form many different membranous organelles. The most famous one is the nucleolus. It's been known since the 19th century. A very big object that produces ribosomes in the nucleus, but there are many other ones, and we're still discovering new ones. So in conclusion, a cell is nothing like a test tube. Nearly everything is organized inside the cell by protein and RNA scaffolds. So emergent properties, very quickly, nearly all biology works by feed forward and feedback loops. I learned when I was a student that A goes to B, goes to C, goes to D, goes to E, which is true, but it's all got to be controlled. Otherwise, it's not useful for cells. And so even with this relatively simple pathway, you can't understand it without mathematics. But here's a much more typical thing. This is the same actin I talked about before, what that major cytoskeletal filament does is carefully controlled by all these other proteins that bind to it, actin-associated proteins. This is just the actin-associated proteins in a yeast cell. Humans would be even more complicated, but here they're colored differently. So, according to what they do, the dark purple ones are involved in filament dynamics. That would be the formins, the one I showed you. There are multiple different formins and other proteins involved in filament dynamics. The famous myosins that cause motion are the green ones, and so on. So even when we know exactly the structure of all these things, their interaction constants, the equilibrium constants, the kinetics, it's just too complicated. We can't, yet predict what will happen to the actin inside the cell because it's too confusing. And so this is really a general problem, not only for actin, but for many other things. So as a consequence of such complexity, we will not be able to understand even the simplest of living cells without some new methodology, because life reflects the emergent properties that result from very complex networks of interactions. So actually, I gave this talk the other day, and my colleague, another professor who was part of this symposium, said he thought I was optimistic. I'm claiming we're not going to really have a true understanding of how cells and organisms work till the end of before the end of the century. He said he thought that was very optimistic. Anyway, we have a lot of work for you guys to do. Much more biochemistry will be needed in purified systems that reconstitute biological systems. And also, we need new quantitative methods for analyzing and understanding the enormous complexity of life's chemistry, new kinds of computer modeling, mathematics. Maybe quantum computing will help, but we need to know what to put into the quantum computer. Okay, third point, fundamental biological research will be essential for improving all these things we need, human health, environment, and so on. Current problem in the United States, me and my colleagues have been criticizing publicly the National Institutes of Health for overemphasizing what's called translational biomedical research, which is very important, but it's not enough. You need a foundation of fundamental knowledge so you could really do the translational work more effectively. And when I was at Science, I had to commission 250 editorials. I wrote maybe 50 myself, but commissioned 200. This is my favorite one from Huda Sogby at Baylor University. She's a very famous and effective translational researcher. She's an MD. She works with children with Rett syndrome, a brain disorder. And she is calling on more young people to work on fundamental research. And she has this wonderful quote at the top. Huda's from Lebanon, so she knows about language. She speaks multiple languages. She says, scientists are trying to translate a text with the sophistication and depth of Shakespeare using a first grader's vocabulary and experience. That's such a great way of thinking about it. I just want to give you a few specific challenges. And maybe when I looked at your program, the next one coming, the synthetic cell, this maybe Spain could take the lead on this. I think we need to get a group of people working on together on some of these problems. And it's frustrating that these wonderfully important central problems are not really being explored. So there was a huge surprise, I think, in 2016 when this group, using very sophisticated methods, were able to make a minimal cell. They started with a mycoplasma and started taking genes out using sophisticated synthetic DNA methods. And to their surprise, there were 473 genes left in order to let this bacteria replicate at once every 3 hours. And of those genes, basically a third of them, they had no idea what they did. This is amazing. So I would contend that, figuring out what those other genes do, there must be Nobel Prizes hidden there, but, it's hard work and it's not fashionable these days to work on those problems. And there's very little work being done on this. This is a huge opportunity to make important contributions to really understand how that cell works. Another example is the importance of Drosophila, the fruit fly, which has been the model for learning how cells work together to form and maintain tissues. Many examples shows that first working on a mechanism in the fruit fly provides a shortcut to understanding humans. As an example, in our last book, our chapter on development of tissues contained 50 references to the fruit fly, 4 times more than to the next most cited organism, which was the mouse. I would contend if we didn't have what we discovered in fruit flies, we still almost know nothing about human development. So consider the human brain now as a fashionable topic to work on. You got to be very brave to work on human brain, especially if you work on the ultimate emergent property, which is human consciousness. It's a great example of what is an emergent property. Out of a network of interactions of cells comes this thing that's completely different, human consciousness. A few people work on it. They got to be very brave. So here's what about the human brain. The human brain contains more than 100 billion neurons, each of which on average has to make connections with 1,000 others according to a regular and predictable wiring plan. So incredible complexity. The Drosophila brain, the fruit fly brain, contains about 100,000 neurons, 1 million times less than humans. You could map every cell. You could make any mutant you want. Or you could get a new generation every few weeks. So much more accessible than the human brain. And I would contend until we understand this brain, we have no hope of understanding the human brain, and we don't even know what to do in many cases. Here, I just want to stress that when I say model organisms, I don't mean only the standard model organisms. We need new models as well. It turns out that cells have something like a GPS system on their surface. They know where things are supposed to be. It's probably an important part of our own development, but it's very obvious in these ciliates, these giant ciliate cells that have a mouth and all kinds of different organs. This is the stentor, a giant single cell that worked on extensively. T.H. Morgan worked on it before he worked on fruit flies in 1900. A man by the name of Vance Tarter spent his life working on it, but it all stopped about 1960 or so. And this young professor, I call him young, and Wallace Marshall had the courage at UCSF to take it up again. But it takes a lot of courage to start this because you don't have hardly any colleagues working on it. You're not going to get many citations. You're not going to get many publications because you have to work out all the new methods and everything. And our system needs to encourage this kind of work because from this system we'll learn how cells know how to position things on their surface, and then now it's going to have major applications for all organisms. That's just one example. And here's the new model organism. Unfortunately, this virus is a complete mystery. It's more than 4 times larger than the flu virus. It produces 29 proteins, most with functions we do not understand. And it's just, why is it so big? What's the advantage to the virus of being so big? We don't even know that. So, but the good thing is it's causing scientists around the world to collaborate and focus on understanding the fundamental cell biology of this virus. And I'm sure that's going to lead to many new fundamental understandings of crucial importance to the future of human health, including a lot about the immune system, which is many of the proteins in these viruses are there to counteract the immune aspects of the immune system, and they're therefore good probes into working, trying to figure out how the immune system works. Again, many mysteries there. Okay, the last topic: how to decide what to investigate as a scientist. There's so many things you could do as a young scientist. Think about it. You got. I want you guys to think carefully before you start. And so I'm going to describe how we're trying to address this question that I've been co-teaching to graduate students at UCSF. And we want students to be ambitious, not just to collect data. We want you to do something important. So what I've been teaching is one of 40 different specialized mini courses at UCSF. In these mini courses, 15 students meet with 2 faculty in 10 3-hour sessions over the course of 3 weeks. So it's a very intensive course. Students read and discuss a series of research publications, and any 2 professors at UCSF can get together and choose a topic. And there's courses on evolution, aging, chromatin, so on. The mini course I've been teaching is called Classical Papers in Biology, and I'm now teaching with Denitsa Fujimori, who's a young chemist, or at least she's younger than me by a lot. And this is the introduction to our course. The point is that we take a series of, I think it's 8 classical papers, pair each one with a modern paper in the same field. But our point is not to teach you history. Our point is to stimulate students to think innovatively about strategies for planning their own thesis research. So we start with Mendel. There's a wonderful, clear English translation of his research 40-page description. And we pair that paper with a modern paper on epigenetics, this paper from Enrico Cohn's lab. We often work with Nobel Prize papers, but not always. But we really like this pair between John Gurdon, who won the Nobel Prize for his work in 1962, with Shinya Yamanaka, who won the same Nobel Prize, shared it with John in 2006. And we encourage everybody and discuss a wonderful talk that Yamanaka gave when he won the Lasker Award before he got the Nobel Prize, explaining his research strategy. And it's it's very well worth watching. And I suggest you search Yamanaka Lasker, you'll find this. Basically, he designed his strategy of how to develop, derive stem cells under pressure to design something ambitious that could attract graduate students. He had just moved to a new university and was failing until then, almost ready to go back to surgery, which is his field. We then do a biochemistry course. We start with Arthur Kornberg's DNA polymerase. We end with single molecule assay paper. We have all other papers that change from year to year, but we always end with this one paper, this one session with Darwin and scientific standards. and we read two chapters from Origin of the Species. And the important thing is we pair this paper with a recent high-profile 18-author paper that has been shown to be completely wrong. It's got a bunch of postdocs and graduate students who were being misled by working on this thing. It's looking for drug targets. And the whole point is how to avoid being misled. Don't fool yourself as a scientist. It's easy to get into that trap, especially when you're working on, in a large group. Why Darwin? Well, Darwin was a model scientist, in my opinion. And here's a wonderful quote from his autobiography. He said, "I had also during many years followed a golden rule, namely, whenever a published fact, a new observation or thought came across me which was opposed to my general results, to make a memorandum of it, to write it down without fail and at once. For I had found by experience that such facts and thoughts were far more apt to escape from the memory than favorable ones." That is, we always can fool ourselves by not paying attention to things that disagree with what we think we're observing. So this is, of course, this is the first year I had to teach by Zoom. And this is my colleague, Denitsa, up there next to me. I didn't know she was taking screenshots, but when I talked about Darwin, I've been to Darwin's house many times, three times I've been to Darwin's house outside of London, and these are pictures from his house, and this is his working desk, which I said I use for inspiration. But at any rate, I think he really is a great model for us all. Okay, our major point in this course is that strategy is everything in science. As students discuss each paper, we dissect the experimental strategies that each scientist designed to attack an important biological mystery. So there are two things. You got to work on something that's really important, and then you got to get a good strategy to work on it. That's what we stress with the students. And we discuss what makes something important and what's not important. And the strategies often involve an investment in developing a new method. That's what I did, of course, with DNA cellulose, and that's what many other scientists did in order to make their work really possible and innovative. So this is the end. What have I learned from a life in science? The central lesson I've learned is science is much more important than most scientists realize. It's critically important that science teachers and scientists achieve a much higher degree of influence throughout both their own nations and the world. I learned my first trip to India, 1993, about Prime Minister Nehru's emphasis on the need for a scientific temper. And that's why he invested in science and technology in his new nation. Every nation needs much more of the creativity, rationality, openness, and tolerance that are inherent to science. And this will require major changes in the goals of science education. And that's another talk, another day. No time for that today. I've been working on that for years, but not with much success. You can see what's happening in the United States right now. We're in bad shape. People with all kinds of magical thinking, it's causing lots of people to die, including our own president. So this is my favorite quote about scientific values. Jacob Bronowski was a physicist. As a young man in the British Army, he flew over Hiroshima and Nagasaki after the bombs were dropped. It was very much depressed him. He said, was science really bad for the world? It developed the atomic bomb. He spent 10 years writing and thinking about writing and reading and thinking about science. And he concluded in the end that, of course, that science on balance is very good for the world. This little book is still in print. It's very worth reading, Science and Human Values. He said the society is simple is because it has a directing purpose to explore the truth. Nevertheless, it has to solve the problem of every society, which is to find a compromise between the individual and the group. It must encourage the single scientist to be independent and the body of scientists to be tolerant. Science has humanized our values. Men and women have asked for freedom, justice, and respect, precisely as the scientific spirit has spread among them. And I'll just end with this wonderful quote from Pasteur, which is very much relevant to my talk today, since I'm talking to you from far away across the world. We need scientists all around the world to work together. This latest pandemic it illustrates the critical role that science plays in humanity, and we need to take advantage of it. We've also learned how far away we are from having a rational, tolerant society in many countries. So there's a huge amount for us all to do. Thank you.
Thank you very much, Bruce. If you could be so kind of stopping sharing your screen.
Sure, of course. Yes. I will turn off my slides. I hope. Trying to get my slides off. Okay, there.
Excellent. There we are. There we are. So if you were so kind of answering the questions that are going to be made by the students of the master, we will be very, very grateful. So the first question is from Sandra. Sandra is here.
I think she isn't, but I can read the question for her.
Okay.
Okay, then I read the first. In your article of 1998, with the same name of your talk, you said, even our most talented graduate students lack the background in the physical sciences that they are likely to need to decipher the detailed chemistry of protein machines. Do you think this has gotten better or worse currently?
I can only talk about UCSF. I think it's getting better, much better. I think one of the, and I only know about the United States, of course. One of our major failures has been the way we teach mathematics to undergraduates. It's taught, you know, at least all the mathematics I took was designed for engineers and not for chemists or not for biologists. And that's because in the United States, mathematics is taught by the mathematics faculty. Today, we have lots of biologists who are mathematics, systems biologists. They should be teaching the mathematics to the undergraduates who want to do biology. Too often they're not, because the courses are owned by the math department. So, you know, universities are not always rational places. For the good of the students, we should be teaching a different kind of mathematics to biologists. What I said in that article, I think, is that you know, our graduate students at the time thought mathematics was irrelevant to their work, even though they had taken it. They didn't see any connection. That was because the way mathematics was taught. So I think it's getting better, but only not as quick. Nothing ever happens as quickly as it should. You know, not everybody needs to know mathematics, but we need people. Anybody who's going to work on the you know, the kind of complex things we're talking about needs to understand both mathematics and biology, because you get people, physicists and mathematicians working on biology, often they're not working on interesting problems. They don't understand what's important in biology. So, you know, if the physicist, anything out there in the university you observe, you could, you know, put in equations and try to explain. Well, but not everything that you could observe in biology is important to put in the mathematics and explain. Only certain things. So you need both mathematics and deep biological understanding in order to understand what's really important to investigate in biology and what to apply all these methods to.
So the second question was mainly focused by Sandra into response that it would be worse, but you said you think it's getting better. But in any case, I wanted to ask the questions about rephrasing it in a way that how the absence of knowledge on this physical or mathematician side could affect the research of these students that are now working. More and more students are working actually at the time now in present times. So how their research could be affected?
Well, I mean, you know, if you're going to work on a problem like that simplest cell, that's a mycoplasma cell with 473 genes. You know, you'd like to be able to figure out, you know, what does it mean to understand that cell? What it means to understand that cell is have a computational model that with all the different protein machines in there, and how they're controlled, and how they interact with each other. That when you alter any one component of that 473 genes, you could predict what will happen to the cell. What will happen to all the other ones when you change one? And, you know, we're really far away from that. So if you're going to work on that, you know, there are too many experiments that you can do. You need some kind of theoretical construct to help guide what your experiments are done. And the theoretical construct's got to involve mathematics and computation. So either you could be working on that problem, but you got to be working with somebody who's, you don't have to do the computation yourself, but you have to be part of a close group that's thinking together. You know, you know, pretty standard stuff to purify the 143 different proteins from that cell that are not understood, and somebody's already done protein interaction networks, so you can start to assemble machines and see what they do. But in addition, you need to see how those machines are integrated. So you need biochemical talents. You need you need people who know how to do all the manipulations to get the proteins out. The problem if you have no connection to the mathematics and computational world at all, then you're going to avoid doing a lot of experiments that need to be done.
Yeah.
And one of the problems today is it's too easy to collect data. So the graduate students at UCSF, I'm trying to discourage, don't just go into a lab and where you could collect lots of data. Data is not understanding. We could know all these facts. It's too easy to collect data. Let's try to understand things. And one of the problems is just trying to collect data. Everybody's doing the same thing. If you're a scientist, you want to do something unique with your life. You only live once. Why do the same thing as everybody else? The easy thing to do is go take some kind of tumor and measure everything in that tumor that everybody else is measuring in another tumor. Well, are we going to learn anything really from that? Are you going to be unique? I see so many people doing that, both at my universities, and I'm very critical of a lot of the research that's being done. That really is just data collecting.
Yeah. Yeah, I think actually you already pointed to the last question from Sandra, that is actually this way to work in an interdisciplinary environment. If you don't know how to do one thing, then you should be surrounded by the people that can do that. I mean, this is hard and this is difficult, but do you think that this is the way to move forward, or the way these graduate students should try to make their path in the research career, finding colleagues that are the experts in the areas where they don't really have an expertise?
Right. I mean, Spain could be the leader in many, areas if you guys would work together. I mean, what the mistake would be to just copy what everybody else is doing in the United States. There's so many things new that could be done. And if you guys could work together, the 12 of you, whatever the number is, you could be a new team. But, I've said this many times, science is too much like fashions, you know. People tend to follow what everybody else is doing, and unfortunately our granting system in the United States does not encourage risk taking enough. My colleague Harold Varmus and Shirley Tillman and I wrote articles about this. We're still working on this. One of the models we like is the ERC Starter Grant, European Research Commission Starter Grant, which funds really new ideas, risky research for young people. We want that. What we want in the United States is that every young person who's trying to start a lab has an equal chance of getting a grant from the NIH by working on something new that they haven't done before, as on continuing what they had done before, which is, so right now, almost all the grants are for continuing what you did before, because they require preliminary data. And of course, you've been on a postdoc, so you can keep on working on the same thing. And we're not getting innovation out of the system. So we would like to switch to a system where half the grants are like the ERC starter grants. But it's hard to convince our colleagues.
I can imagine. Thank you. Thank you.
Now you have the microphone, Mateo, if you want to deliver your question.
Hi. Yes, my question is kind of linked to the last slides in your presentation, in which you said science should have a higher degree of influence. So it would be, what role do you think science should play in politics, both in a broader sense, such as aspects that could be applied to the political process itself, and in a more specific way, in the issues kind of way, like global warming or public investment in research, which you have just mentioned? So what do you think the relationship is between these two?
I could talk for many hours on this topic. So first of all, we have to change science education so our politicians understand what science is. Our politicians get away, and the public has to understand what science is. Politicians in the United States could get away with saying, at least in some states, climate change is a hoax created by scientists to get research grants. That was said by the governor of Texas, and then he became Secretary of Energy for President Trump. You know, and he could never say that if people actually understood how science worked. Then you look at how we're teaching science, and you see why it's possible for him to say that. We teach science as if it's important for our students to know all the facts that science has discovered. I would think that by now I realize it's much more important that they understand what science is and how it works and why they should pay attention to science. You know, why does a scientific consensus on smoking or climate change or on wearing masks, for example, in the United States is, you know, 1/3 of the people refuse to wear masks. And the internet, there's all this crazy stuff that masks cause disease. I mean, just crazy ideas going around that are killing people. And I think it's partly our fault. Those of us who teach science have to change our focus and make sure that every time a student gets at least a C grade in our science course, they at least understand how the scientific enterprise works. How does science work? We don't teach that. You can't tell them. You have to give them examples. They should read a scientific paper, and we should discuss how science works and why it's impossible that scientists could have created climate change to get grants. I mean, and they need to understand why you could always find a scientist who will disagree with anything, because you get to be famous as a scientist if you show that the consensus is wrong. But that's the way science works. And they need to understand why scientists can never say, I'm 100% sure that human activity causes climate change. They'll say, I'm 99.9% sure, because we have to be open to being disproven. And the other side, you know, there are all these people who know nothing. They're absolutely sure that climate change is a hoax. And so they have debates with scientists, and the scientist is acting scientific, and the other person is acting like some kind of dictator, and the public is confused because they don't understand how science works. So, we have a huge problem. I mean, the first thing is to get the politicians to understand what science is and why they should respect its judgments. And then we could interact with them much more effectively. And the other thing I would say is, I know Spain has a young academy. Has anybody heard of a young academy?
No.
Spanish Young Academy?
Yeah, it is known.
So I'm part of the old academy network. The standard academies have been around for hundreds of years, some of them, and we have a network trying to work on problems, but we're not on social media. We're all not connected to the world anymore. The Germans in about the year 2000 formed a new invention called the Young Academy. The old academies had this brilliant idea, very risky, to form a Young Academy. They appointed, I think, like 100 Germans, scientists, engineers, and political scientists too. I mean, a mixture of people, all aged 35 to 40, to be part of a Young Academy that only lasts for 5 years. I mean, they're in the academy for 5 years, and then they graduate, and then new people come in, 20 new people every year. So it's always a Young Academy. It's not part of the old academy. It's just encouraged by the old academy. And the important thing is, if you're a member of the Young Academy, you have an obligation. You accept an obligation to do something for your nation. And the other second important thing is the old academy doesn't tell what the Young Academy what to do. Each Young Academy class thinks of 1 or 2 projects they want to pursue. And this has turned out to be an incredibly effective way to mobilize science in a country. I was science envoy to Indonesia, and I was there while they formed a Young Academy. And there are now 40 countries with Young Academy. The United States is about finally to have one. Spain has one, I'm told anyway. They're all over social media. They're much more effective than the old academies. The other thing that's really relevant to the question is that in Germany first, but now in Indonesia, I see it very clearly. The politicians want to interact with the Young Academy members. They don't want to interact with the old Academy members. The old Academy members remind them of their chemistry professor who gave them a C or a D. Older scientists are threatening to politicians or have bad reputation. You remind them of something bad from school. This is my theory anyway. But the young scientists remind them of their children or their grandchildren. They're not threatening. And so in both Germany and Indonesia and in other countries as well, the political leadership wants to meet with the young scientists, and it's been a big surprise, but it's now been shown over and over again. So I hope the Spanish Young Academy is doing well. I know the Indian Young Academy has 100 members, and it's doing really well. The U.S. just started with 18 members, the first class, and we're, I think, about to take a second class. I could not get my successor at the academy to do this. So for 12 years, we couldn't do anything. But now Marcia McNutt, who's the new president, she's very enthusiastic, and we're going to get a young academy off the ground. Now, all the young academies are networking around the world. They have regional meetings, in European young academies all get together once a year, at least. The Asian young academies. Anyway, I'm a big fan of the young academies, and I think that because they're much more, much less threatening and much more interesting to politicians, it's a great way to get science into the political system.
Thank you. Noelia, you have to switch on your microphone in order to make your question.
Hello, good evening. Thank you so much for your talk. It was really inspiring. The question I prepared, you have already answered it with the previous question, because reading the article and your webpage, you center so much in physics and mathematics. Because of my short experience in research, or because I remember one of my professors in the university, and also thank you to Herman. I was thinking about an integrative point of view to do the research, because it's great to know the speed of one enzyme or knowing its concentration, but if you can't relate it to the whole cell or the whole process, I think you lack a whole bunch of information that it's needed to understand that type of process or what you are researching. So I think maybe that's my personal interest. Uh, it's key to know all those mathematic things, and without them you cannot make statements about that protein. But I like to connect all these little things, that little features of each proteins, with a complete process that lead to something like replication or cell death or a pathology. So that's something I was thinking when I read your articles and throughout the talk. So you already answered it. Thank you.
Well, you mentioned apoptosis. Well, which, apoptosis is very important for cancer, because cancer cells evolve to not kill themselves. Cancer cells are very much out of whack, and they're disturbed, and they would have triggered. They should have killed themselves already, because all these fail-safe mechanisms that evolved to make multicellular organisms possible, use the cell suicide to protect us from cancer. So all these cancer cells have distorted their apoptotic system, so it's critical that we understand how that network works. And so before we understand the whole cell, of course, we're going to have to work out methods to really understand parts of it, and that would be a great focus, for example, because it's so important. And it's not nearly as complicated as the whole cell. So be able to compute by taking a cancer cell and see what its apoptotic system is thinking. What is it? Why is it not killing itself? What has changed about it? Why is it? How is it set? And if you understood how that network actually worked, you then could make that cell kill itself, presumably. So there's a great problem too. I mean, that problem is being worked on, but usually the people who are doing the biochemistry are not interacting with any mathematicians or computer scientists, and it's slowing down the success of that work and actually getting to the answers we want. So I think one of the things we've learned from this this horrible pandemic is that we could use Zoom much more effectively to connect scientists than we did before. There's no reason why, I couldn't be having if I'm working on apoptosis, create a group of, 5 people from around the world with different skills, and we could meet every Monday at 10 o'clock by Zoom for an hour and show data. And, there's new ways of thinking about science that are less isolated. Everybody's got their own little thing and thinking only about themselves. We need to do experiments about getting the right people together. CSIC should be doing it, and do experiments how we could use our new willingness to think that Zoom and the chat screen and all that stuff really make scientific meetings pretty effective, even when you're not together. So, and you could do it every week for an hour, or once a month for 2 hours or something. I think we need to make science better than it is by taking advantage of what's happened with our new culture. And I mean, I've been surprised to attend scientific meetings that have been very effective, even though they were on Zoom. Things I would never have thought could have worked. And I think that we should have a chat box, chat feature for every in-person seminar, because there's a record of all these people are interacting with each other while they're listening to the talk. Then you can record it and try. I'm the chair of a scientific advisory committee for something called the Scleroderma Research Foundation. We had our annual 2-day meeting by Zoom because we couldn't meet, but it was incredibly effective in connecting people. And I was really surprised. And I think we've all been surprised how much we can do without always having to be in person with the same person. And so, for science, it's really exciting to think about what we can do to create new ways of joining together so that people working on related problems can help each other.
Thank you. Miriam, you have the microphone now, please. You have to activate the microphone.
Okay, yeah, I'm ready. Thank you for your speak. Good afternoon. And some of these questions, you already mentioned some of them, but I wanted to ask you, what are some of the best qualities scientists should have, and what kind of profile is most appreciated in prestigious universities in USA or wherever? You already mentioned we have to be patient, and we would like to know more qualities.
I'm sorry, you'd like to know what? Could you repeat?
Yes, what are some of the best qualities scientists should have, and what kind of profile is the most appreciated in prestigious universities?
What do you mean for thinking? You're thinking about if you work with somebody in the United States, what should you look for?
No, like what you will ask for us, like what you would like us to have as qualities in order to be accepted in an university or in a group of work or wherever.
Well, so what you could ask. What do I look for in a young person?
Yes,
I can't that I could answer. I don't know. So I think the wonderful thing about science is that scientists is all about what you're thinking and what you're saying, and not who's saying it. So when I look for a student who has shown me some deep thinking about anything. Generally I'm most impressed by people who have thought about something or done something that I had never thought about, or that I think I might not have ever done. You always look for people who have a spark of creativity and depth of thinking. Not, I don't care how many techniques whether you've done this or that techniques. That's I would call it the plumbing part of science. It's how you think about science and what's in your head. I recommend to students this website that my colleague developed called iBiology. Have you heard of iBiology?
Sure.
iBiology is a free website with like 1,000 different scientists talking about their science, and I recommend that young people, when they're tired and have nothing else to do, go listen to some of those talks, because what you need to get in your head is a lot of different examples of ways people have solved problems and thought about their problems, and then be able in your to come up with some combination of approaches, of skills, techniques to do something different. So I look for young people who are thinking, you know, interested in an important question, and have done enough reading and thinking to tell me something interesting about it that I hadn't thought about. You know, so I'm not looking for young people who know how to do PCR or this, that, and the other thing. I want to see them, you know, that they have thought deeply about something and invested time in reading and thinking, because that's what it takes to be a successful scientist. I've often said that, you know, good science is like good art. You know, and I've made the analogy with giving me all of Rembrandt's paints and materials, and having me paint a painting. I would come up with nothing useful, and most people would come up with nothing really great. Rembrandt had a great talent of combining things in unique ways to make a great picture, and other great artists have that ability too. And this is exactly the same thing about science. You have all these methods. You don't have paints. You have methods and techniques and approaches, and you know, and you got to mix them in a way that makes great science. And it's equally creative to be an outstanding scientist as to be an outstanding artist. And I think that analogy helps to emphasize what's really important about science, is thinking about something differently, coming up with a strategy that, you know, is interesting and different, and not doing the same thing as everybody else, and shows that you are trying to solve a problem that really will make a difference if you solve it. I mean, there are lots of problems in biology that are not interesting. I mean, to figure out exactly why some cancer cell is doing something crazy may not be interesting, but it may not tell you anything. Other mysteries are very important, like the one I talked about. Why is a cell alive with 473 genes? What does it mean? You know, a cell could reproduce itself. Chemically, it's a collection of catalysts that makes more catalysts just like itself. So it's a self-replicating collection of catalysts. That's what a cell is. And we can't really explain it. We don't have we couldn't even predict what genes would be essential and what wouldn't be essential. They had to do it by hit or miss. And that's what all that work was about. And they came up with this result that nobody expected. Most of the 1/3 of the genes, we don't even know what they do. So that is an important problem. What makes a simplest living cell is important. Many other things are important, but there are lots of things that are not important. So I think when a young scientist, I look for you know, interesting thinking, evidence of having done enough reading and thinking that, you know, they know something I don't know. That's the kind of thing I would look for.
Okay, thank you very much.
Thank you very much. I think we're running out of time. I'm going to pass one more question. Gianfranco, are you ready to make your question? Open the microphone, please. And then
Yes.
we will pass on the microphone to Dr. Germán Rivas to close the event.
Okay. Hello. My question is a bit more maybe philosophical. I've heard you say that instead of learning facts, we should learn how to think. And I have this question on how can our teachers teach us how to think rationally, and what can we do as students on how can we work on this idea of thinking as a scientist?
Well, you know, there's a lot to say about science education at the college and graduate school level. You know, we used to think that just talking to students, giving lectures was teaching. I don't think that's teaching anymore. It's not enough. We need to let students struggle with problems and try to solve them, and work together in groups. We know a lot about how to do science education from a lot of research on science education. And we know that, I'm a professor because I sat in at lectures and I was good at learning from lectures. And so therefore, as a professor, I think that everybody learns from lectures. So all the professors at my age were selected because we were good at learning from lectures. But we now know that there are people who are not most students learn better by struggling with problems and working with colleagues, not one-on-one. And so even, our academy has done a lot of work analyzing the research that's been done on college science teaching, and there's a free book. Our academy has a website with all the 10,000 publications that the National Academy of Sciences of the United States has prepared on any topic, how much arsenic in drinking water is safe, what's happening with climate change and all that stuff, but also on education. And there are books, there are free books aimed at college faculty explaining what we know from research about how to teach best, and it's using active learning in addition to just talking. And the book you want to search for, if you go to National Academy Press is where the website of all the books are, 10,000 books, and you search for, there's a book is called Reaching Students. It's aimed at college faculty. It tells examples of college faculty changing their teaching and learning from, you can't change all at once. You can change, try changing for a week and then 2 weeks. And anyway, we know a lot about what you're talking about. The other thing I would say, you have to fail a lot. You have to learn from your failures. So every time you fail, write down, think carefully about what you did wrong. Write it down and say, what am I going to do differently next time? What will I, what did I learn? I would say a good way for students to learn is to write down, once a week, write down what you're thinking. I find that by writing, and that's it helps me think. So write down, I've done my experiments. What I think about my experiments in the last week. And start writing. You start thinking differently and more deeply. The mistake is just to go on long without thinking, and that's really bad. Keep doing the same thing and don't think. You got to make yourself think.
Okay,
thank you very much.
Thank you. I'm going to pass the microphone to Dr. Germán Rivas, who will now follow up the discussion.
Hello. Unfortunately, we don't have time to questions from the audience that had been following the talk on the YouTube channel, and we thank all of them for their presence. But we are going to send Professor Alberts a list of the questions that I think some are very interesting, and perhaps you can think and you can make a kind of homework in San Francisco, because it's been very active and we are getting questions from Chile and on very different topics, including your statement on coronavirus as a model system. Many interesting questions, and is being active. And if you don't mind, many of the questions from the students have not been possible to talk about them now. If you don't mind, after the YouTube is closed, perhaps we can have like 5 minutes to get together and say goodbye to you in a private way in the Zoom, I guess. But let me say a few words to conclude on behalf of the entire team of the Master in Molecular and Cellular Integrative Biology, MCIB, in which with great enthusiasm and effort, we are making possible this advanced training program as an in-house activity at the Research Council centers. This event has been a lesson with capital letters, delivered by a master with capital letters too, addressed mainly to our future generation of molecular cell biologists that here are represented in the Zoom room by the MCIB master students, but others are listening from other places. Thank you, Professor Alberts, for accepting our invitation, and primarily for your outstanding long-term contribution over decades to educate a worldwide creative minority, particularly in biochemistry and molecular cell biology, and more generally in scientific thinking. In this regard, the textbook you first authored are landmark examples. Plato, I'm going backwards, remind us, the philosopher Plato, remind us that the teacher's voice is much more decisive and crucial than any book, including yours. And I'm confident that your voice, Professor Alberts, in today's talk will be decisive or crucial for the future in science and life of a significant fraction of the young audience that has been listened to you today. Dear students, as senior scientists and teachers, we will try to do our best to help you keep the momentum. And if you finally decided to continue exploring the unknown, becoming a scientist, please don't forget that science is more than just a profession. It is a way to look at yourself and the world around you, to solve problems that matter what the answers are to science generally or the humankind. Thank you, the Research Council and the Menendez Pelayo University for the organization of this master. Thank you, all of you, for your attention, and especially thank you, Professor Alberts, with our deepest gratitude.
Thank you. It's been a pleasure. Good luck to all of you. We're expecting big things. We need it.
Thank you very much.